Hello everyone, this is our first AP Biology video. I want to go over the rest of Chapter 3 with you so we don't have to spend any more time in class going over these material, and you'll have a better idea on what's the most important knowledge that you're supposed to get out of this chapter. So for our orange class, we haven't finished the protein portion, and for our blue class, you can move on to the next page when we get to it. So this is for our orange class. We talk about primary structure, secondary structure, tertiary structure, and quaternary structure of protein. So we have these notes that go along with it, just in case you ever need to come back. Well, you should at some point come back and review it when you forget what they're about. So our primary structure is determined by DNA sequence, right? DNA, RNA, protein. So when we have our amino acid chain, those are all determined by the DNA sequence, how the amino acids are combined, what sequence they go in, is determined by DNA. If you change one amino acid, it may or may not have have an influence on how the protein is folded and how the protein functions. So sometimes when you change one amino acid, there's a chance that your protein might still be fine, but sometimes, for example, in the case of sickle cell disease, when you change one amino acid, it could completely screw up the shape and the function of a protein, right? We always talk about structure and function. When you mess up the structure of a protein, the function of the protein could also be influenced. So in this case, for sickle cell disease, when we have our normal red blood cell supposed to have protein in the red blood cell that allows transport of oxygen. However, because of this one amino acid change, the protein is not folded correctly because the interactions between the amino acid is now messed up. And now we have a protein that doesn't carry oxygen as easily, and therefore we have sickle cell disease. Second one is our secondary structure. There are two types of secondary structure. There's the alpha helix and beta sheet. As you can see, there are a lot of hydrogen-oxygen bonds, so that's our hydrogen bonding that happens in between each one of these amino acids. So if you look right here, this is one amino acid, but then further down the chain, there's another amino acid that can interact with this amino acid in order to form these hydrogen bonds. So those hydrogen bonds are formed because of the primary structure of the protein. So this has nothing to do with DNA anymore, right? DNA did its job when we form our primary structure, but how is the protein folded is completely based on the sequence of amino acid and the property of each amino acid. So here we have our secondary structure. There are these two types, which are both stabilized by those hydrogen bonds. And the next one is our tertiary structure, which is made because of the interactions between the R groups. So right here, we're not really talking about the interactions between the R groups yet. So if you forgot what the R groups are, you see right here one more time, for every single amino acid, you have an amino group, you have a carboxyl group, and then you have the R group. So each one of these is an R group. And because each R group looks different, they have their special properties. They could be nonpolar, polar, electrical charge, acidic, basic, depending on whether it's a negative charge or a positive charge. But even within, let's say, all of the nonpolar amino acids, since they're still different chemical, right? The R groups are still different. Each one of them perform a little bit differently as well. This is why we can't 100% predict how a protein is folded just yet. So tertiary structure is caused by the interactions between the R groups. So for example, over here, you can see this is this is a polypeptide chain, I guess. It loops around, I guess. So we can have the interactions formed between the R group of different or even same amino acids along the chain. So there are several interactions that happen. Basically, any kind of chemical interaction could result in the formation of some sort of bond that allows the specific folding of a protein. So The first one is polar interaction. That's when you have two polar molecules. We don't have a full charge. It's when you have a partial positive charge on one R group and a partial negative charge on another R group of another amino acid. They interact with each other. That gives you your polar interactions. The next one is ionic bonds between the charged R groups. So if we have a positive and a negative R group, they can form ionic bonds. And then we have hydrophobic interactions. If our R group is hydrophobic, it would tend to fold to the inside of the protein, right? Or if the protein is interacting with the hydrophobic tail of cell membrane, then that portion of the protein needs to be hydrophobic as well. So that's what I drew on the board the other day. If you can't remember, please come see me and I'll explain to you one more time. The last one is our van der Waals interaction, as well as hydrogen bonds. So more hydrogen bonds can form, but you can also have van der Waals interaction, which is caused by the fleeting charge of generally nonpolar molecule. The next one that we've never talked about before is called a disulfide bridge. So a disulfide bridge is formed between two sulfur groups, between sulfohydro, so SH, one SH can interact with another SH. So just keep in mind that this is something that exists. The last one is our quaternary structure. For example, collagen is made out of 4 subunits, right? So quaternary structure is when you put multiple proteins together, and those multiple proteins interact with each other and form a larger quaternary structure, and that allows all the proteins to work together and function as a whole unit. So then this quaternary structure right here has 4 subunits. And when we talk about our DNA wrapped around histone, in order to form chromosome, histone protein is also formed by 4 different proteins. So this picture shows you one more time our primary structure. So these are all symbols that we use to represent the different amino acids. Remember, there are 20 different amino acids. So this is our primary structure. It's just a chain of amino acids, polypeptide chain. Secondary structure, you have parts of the protein forming alpha helix, parts of the protein forming beta strands or beta sheet. And then tertiary structure, you can still see all those alpha helix and beta sheets, but they interact even more in order to form into a completely folded protein that's supposed to function correctly if it's folded correctly and it has the correct sequence of amino acid. And the last one is our quaternary structure, where you have multiple subunits. So the next one is, we talk about protein folding a lot, but it's not something that, well, it kind of happens magically. There's a lot that goes into helping protein folding. So now we want to talk about what exactly allows protein to fold correctly, right? It's unlike if you were to fold an origami. You have the instruction, you can fold it right, but you as a person have to do the job to fold it right. For protein, it's the same thing. It has to be in a certain environment in order for the protein to fold correctly. So the first one we want to talk about is pH. Depending on, so you're a pH scaled, whatever pH that the protein is in will determine whether a protein can fold correctly or not. If the protein goes into an environment where the pH is incorrect, then it will not fold correctly. So during the weekend, you should have studied the macromolecules, right? Well, not macromolecules, sorry. The organelles. So we have, let's say, the rough ER and the Golgi. Those are locations where proteins are made in the rough ER and is folded in the rough ER. Within those cysteinae, those little discs of the rough ER or the Golgi, there's certain chemical environment that allows protein to be folded. So pH and temperature within those folds of the rough ER allows protein to fold correctly or not. The next one is called denature. So if you were to have a protein and you transfer the protein to a nonpolar solvent, then all those interactions between each one of the R groups will not happen. So that's a problem. If you change the pH too much, your protein also denatures. So denature means a protein unfolds. If a protein unfolds, it will go back to this state or this state, and then it's no longer in this state, which means the protein is no longer functional. And also, protein could also denature under high heat. Some proteins can return to its functional shape after denaturing, which is kind of interesting. So you can unfold the protein because of an environmental change, but the protein can possibly fold back into its correct shape again. You definitely 100% need to know that the amino acid sequence, which is determined by the DNA, is the instruction for protein folding, right? We call this a spontaneous process. I'm going to write this word right here because it's so important. Spontaneous. This whole protein folding process is a spontaneous process. It can happen on its own, only based on the amino acid sequence. So here we have chaperone proteins. The chaperone proteins are these outside little shells. Those chaperone proteins allow polypeptides to go in there and allow it to fold correctly as well. So just something that you might be interested in. The last one is, how do we know? There are ways that we can learn about proteins. So here are some ways that you can learn about proteins. Now, moving on to nucleic acid. Nucleic acid obviously is related to genes. A gene, once again, is a sequence of DNA that codes for a protein or a trait. A sequence of DNA is talking about a sequence of nucleotides. And we talk about the name of DNA and RNA already, and we know that the monomer of a DNA is nucleotide, and a chain of nucleotides. It's called a polynucleotide. We don't really use this word, so you can ignore it for now. Here's a comparison chart between DNA and RNA. DNA is always double-stranded. RNA is usually single-stranded. It says generally single-stranded, but occasionally there could be cases where RNA becomes double-stranded at a certain part of an RNA, and we'll talk about that in the future when a scenario comes up. So DNA has a deoxyribose as the sugar. So this is a part of your DNA nucleotide. Remember, a nucleotide for DNA or RNA all have a sugar. It should have a phosphate, a P with a circle that I drew usually, and then over here you should have a base, right? So here we have deoxyribose as sugar, and then ribose as sugar for RNA. So that's the difference between deoxyribose and ribose. We're talking about one oxygen difference on the sugar part of a nucleotide. So a nucleotide is one unit of a DNA or RNA, right? But if you break it down even more, we have sugar, phosphate, and base. And then we look at bases that are being used in DNA or RNA. There are 4 bases in DNA, which are A, T, C, G. A matches with T, C matches with G. So when you have one strand of DNA that has A right here, on the other strand that's matching with the A is supposed to be a T base. But since the bases are kind of representing what kind of nucleotide it is, so that's when we say A matches with T, it's really the base of the A of one nucleotide is matching with the T base on another nucleotide on the other strand of DNA. For RNA, we have A, G, C, U. A matches with U and C matches with G. If an RNA were to become double-stranded, obviously the A is going to match with U and C is going to match with G. But also, if you were to have a DNA being transcribed into an RNA, the T on a DNA will be matching with an A on an RNA nucleotide, right? So we'll talk about this when we talk about genetics in a few weeks. But what you need to know is that, let's say there's a DNA that has a base of A, for example, and we're making an RNA nucleotide that comes over and matches with the A would be a U. The next one is function. There are a lot of functions of DNA, right? There's storage of genetic material. There's allow living organisms to reproduce, allow traits to be expressed, all of that. DNA provides direction for its own replication. So what that means is when we have one DNA sequence on one strand, that allows this entire DNA to be replicated. So we can go from one DNA strand and it can completely replicate itself and make another DNA strand. The reason for that is because of the base pairing of the A, T, and C and G. DNA can code for RNA, that's our transcription. There are different types of RNA. Just write the last one. All right, there are different types of RNA. There's mRNA, which stands for messenger RNA. There's tRNA, which stands for transport RNA, and there's rRNA, which stands for ribosomal RNA. So starting with the mRNA, it codes for a protein. It directs the ordering of amino acid into a polypeptide. So that's when we talk about DNA, RNA, protein. We're really talking about DNA, mRNA, protein. So mRNA is the messenger. It sends the message from your nucleus to the cytosol in order to allow proteins to be made, in order to allow traits to be expressed. The other type of RNA is called transport RNA, tRNA. So tRNA carries one amino acid by matching the codon with the amino acid. So what does that mean? One tRNA is an RNA, obviously, but this RNA carries an amino acid with it. And when we're making a protein, these tRNA are responsible for delivering the amino acid in order to make a polypeptide chain. Once again, when we get to our genetics unit, we'll look very specifically into all of these molecules again, but right now, just know they exist and they're different types of RNA. The last one is rRNA, ribosomal RNA. If you studied over the weekend, which you should have, rRNA is a part of ribosome. It's something that allows ribosome to function. There are other types of RNA as well, but we don't really go into those in our class. DNA, RNA, protein, there's another name for it called central dogma, because that's the central idea on how everything is expressed and allow living things to be living, DNA, RNA, protein. So you should always definitely know this. This is so simple, you have to know it. Ribosomes are located at two different places. You can find them on the rough ER specifically. If you're looking at a eukaryotic cell, remember for a prokaryotic cell, they do not have a membrane, right? If they have a cell membrane, but they do not have the intermembrane system, so they do not have a rough ER. So they only have ribosomes in the cytoplasm, which means it just kind of flows around in the liquid in the cell. But even for a eukaryotic cell, we still have ribosomes in the cytoplasm. So once again, ribosome can be found at these two places. However, if you're looking at a prokaryotic cell, it will not show up in the endoplasmic reticulum because a prokaryotic cell does not have an endoplasmic reticulum. mRNA has to travel from the nucleus to the cytoplasm to make protein. Okay. So right here it shows you one more time. Oh, here we go. We have a DNA, sugar, phosphate, base. Sugar, phosphate, base. This is one nucleotide. This is another nucleotide. So this picture shows you 1, 2, 3, 4 nucleotides, okay? And as you can see, for each one of the nucleotides, they look different. They have different chemical properties. That's what allows the A to match with the T, and then what allows the C matching with the G. Another thing that's really important is, if I were to ask you, is it the AT bond that's stronger, or is it the CG bond that's stronger? The answer will be C and G, because you're forming 3 bonds right here instead of 2 bonds during the interaction. One other thing to notice is we have, for each DNA strand, remember DNA is double-stranded always, for each strand, there's a 3' end and a 5' end. So the 5' end, we call it the 3' OH end and 5' phosphate end. Okay, 3' OH and 5' right here. The 2 strands of DNA are always the opposite from each other. So if this end is 3, this end has to be 5. That's because that's what allows the DNA matching, nucleotide matching to happen. DNA has 3 parts. We already talked about this. 2' end. Oh, okay, so the 2' end about the hydrogen and the OH is talking about right here. So this is our 2' end that's attached to a H or a OH. So you just know that that's one difference between DNA and RNA. They have their sugar is different. No, we're almost done. Yay. 3' OH, 5' carbon, double helix, anti-parallel. So when we talk about this has to be 3, this has to be 5, that's what the anti-parallel is talking about. The 2 strands of DNA are parallel, but the direction on which way it's going, 3 to 5, and then 3 to 5 this way, is anti. It's the opposite, so anti-parallel. 2 strands of DNA are connected by hydrogen bonds between complementary base pairs. So these are all hydrogen bonds, right? Remember I said hydrogen, it doesn't have to be a hydrogen oxygen interaction in order for a hydrogen bond to be formed. A hydrogen-nitrogen interaction could also allow a hydrogen bond, because the hydrogen is right here is very positive, well, very partial positive, and then the nitrogen right here is very partial negative. And then we have RNA.